So we are moving on to Unit 7, Enzymes. So this is the structure of an enzymes. Actually, enzymes are what? Protein, and which is made up of amino acids. So in a protein, the amino acids are arranged in different configuration. As you already studied the primary structure, secondary structure, you can see the secondary structures like coiled forms are there, then beta helix will be there. So this highly coiled form is called what? Uh, Tertiary structure. If it involves more than two proteins are there, then it is called a quaternary structure. Okay, so this is actually look like a protein or an enzyme. Now, what are enzymes? Enzymes are actually biocatalysts, and the term was derived from the Greek, which means yeast, in yeast. Actually, the first enzymes was derived from the yeast, hence the name enzymes. So it was introduced by the scientist called W. Kuhn in 1878. So enzymes present in almost all cytoplasm, mitochondria, tissues, and body fluids, etc. Named as enzymes present in the saliva. സലൈവയിലേക്കാണ് enzymes ചെയ്ത salivary amylase. So almost all body fluids contain enzymes. So what are their functions? They speed up the biochemical reactions without being changed. They are not changing at the end of the reactions. So enzymes are proteins that increase the rate of reaction by lowering the energy of activation, which will be discussed later. The energy, what is the energy of activation, will be discussing in the later part of the class. Is a very important topic, energy of activations. Not altered or consumed during the reactions. The one of the important character of enzymes are they are not all altered. The structures are not altered and they are not at all take part in the or consumed in the reactions. They are also reusable. It can be used again and again. Now the classification of enzymes. How the enzymes are classified. The enzymes are named based on the substrate reactions, based on the substrate, reactions, synthesis, chemical nature, all these four important things on which the enzymes are classified. And how do an enzymes are named? The name of an enzymes in many cases end in ASC. For example, you heard about the substrate called sucrose. Sucrose. The enzyme which converts sucrose is called sucrase. So what is the last word? Is ASC. ASC is added. So the substrate can be converted to its corresponding enzyme by adding what is called ASC, sucrase. If it is lactose, then it is lactase. Likewise, the name describes the function of the enzymes. For example, oxidases, which means what? Oxidation reactions. It catalyzes the oxidation reactions. Sometimes common names are also used, particularly for the digestion of enzymes such as pepsin. Pepsin is catalyze the peptide bond, and also the trypsin. Both the trypsin and pepsins are protein digesting enzymes. So they are commonly used such that. Some names describe both the substrate and the function. ചില എൻസൈംസിൻ്റെ പേര് കിട്ടിക്കഴിഞ്ഞാൽ നമുക്ക് രണ്ട് മനസ്സിലാവും ഏതൊക്കെയാണ് ഏത് സബ്സ്ട്രേറ്റിന് മുകളിലാണ് അത് ആക്ട് ചെയ്യുന്നത് എന്താണ് അത് ചെയ്യുന്നത് അതിൻ്റെ ഫംഗ്ഷൻ എന്താണെന്ന്. ഫോർ എക്സാമ്പിൾ ആൽക്കഹോൾ ഡീഹൈഡ്രോജിനേസ് oxidases ethanol. Ethanol is an alcohol. So the substrate is what? Substrate is alcohol. And what is the function? Function is dehydrogenase. Dehydrogenase, removal of hydrogen atoms. Okay, so both substrate and functions are involved in it. Alcohol is a substrate, dehydrogenase is the function. Now, internationally accepted classification of enzymes is that enzymes are classified into 6 functional classes. It's a very important one. It's called EC number classification. Shortly, it's called what? EC number classification by the International Union of Biochemists, IUB, in 1961 on the basis of type of reaction that they catalyzes. So EC1. is a uh, enzyme. Classification number 1 is the first enzyme is called oxidoreductase. Second enzyme is transferase. Third is called hydrolysis. Fourth is called liases. Fifth is called isomerases, and sixth is called ligases. So EC6 means ligases, not liases or isomerases. So this is a code number. So it should be correspond to exact enzymes. EC3 means what? Hydrolysis. EC2 means what? Transferases, not oxidoreductase. So EC1 is always oxidoreductase. It's a short form OTHLIL. Just to remember. ഓർമ്മിക്കാൻ വേണ്ടിയിട്ട് ഈ ഓർഡർ ഒന്ന് ഓർത്ത് വെച്ചാൽ മതി. Now take the first one, oxidoreductase. So what is the function? It involves oxidation and reduction reactions. Oxidation and reduction reaction. Oxidation means what? Addition of oxygen or removal of hydrogen atoms. Reduction means what? Addition of hydrogen or removal of oxygen. And what are the important subclasses of oxidoreductases? There are many subclasses under oxidoreductase. So dehydrogenases, the name itself suggests that it removes the hydrogen from one substrate to another. It catalyzes the removal of hydrogen from one substrate to another substrate. For example, this is a substrate, AH2 is a substrate, and B is another substrate. So here you can see the hydrogen atom, hydrogen molecules are there. So this enzyme, dehydrogenase enzyme, removes this hydrogen atom and transfer to here. So AH2 become A and B become BH2. So the hydrogen atoms are removed, deleted from here and added here. So this is called a dehydrogenase. So one of the example for alcohol dehydrogenase is alcohol dehydrogenase. So this is malate or malic acid. We can see two hydrogen atoms here. So malate dehydrogenase. So what will they do? Malate dehydrogenase means another name of alcohol dehydrogenase. Removes these two hydrogen atoms and the product become oxaloacetate and transfer to NAD. NAD receives the hydrogen atoms and becomes NADH and H plus. Clear? So these two hydrogen atom are removed by alcohol dehydrogenase and transferred to NAD. So this is one of the example for alcohol dehydrogenase. So this is the first subclass of under oxidoreductase. Next one is oxidases. Oxidases means catalyze the removal of hydrogen from a substrate and pass it on to oxygen atom. It gives to oxygen atom. Hence it is called oxidase. തൊട്ട് മുന്നേ പറഞ്ഞത് വേറെ സബ്സ്ട്രേറ്റിലേക്കാണ് അത് ഹൈഡ്രജൻ ആറ്റത്തെ ട്രാൻസ്ഫർ ചെയ്യുന്നത് ഡീഹൈഡ്രോജനേസിൽ. ഇവിടെ ആ ഹൈഡ്രജൻ ആറ്റം ട്രാൻസ്ഫർ ചെയ്യുന്നത് എവിടത്തേക്കാണ്? ഓക്സിജനിലേക്കാണ് ട്രാൻസ്ഫർ ചെയ്യുന്നത്. So same product is called 2AH2, just a representation. Oxygen atom is there, then this hydrogen atom is removed to oxygen. So this becomes water and this becomes 2A. Clear? One of the examples is called cytochrome oxidase. Cytochrome oxidase is a respiratory enzyme, very important enzyme, cytochrome oxidase. And third one is the oxygenases. Third category is called oxygenases. There are two enzymes as under that, peroxidase and catalysis. So what they do, which are involved in the transfer of oxygen molecule to a substrate. Transfer of oxygen molecules to a substrate. So dehydrogenases and oxidases transfer hydrogen atom. Here oxygenase transfer what? Transfer oxygen molecule to a substrate. The reaction oxidases the substrate by transporting oxygen from molecular oxygen to the substrate. So these are some of the example for what? Oxygenases. Is the transferases. Very simple transfer of a group from one substrate to another substrate. So A is is a substrate and X is the group, B is another substrate. So these enzymes transfer this group to B. So the AX becomes A and B becomes BX. So this is not a hydrogen. Here it's not refers to as hydrogen or oxygen. This is a group. Okay, for examples, transferase or examples are transaminase or enzyme. Transaminase. So which transfers amino group from alanine to alpha ketoglutarate. So this is alanine. So this is the subgroup. So this is another substrate. It's called alpha ketoglutarate. Transferase is what? Transaminase. Transaminase. Here it is transferase. What it transfers an amino group. Hence called a transaminase. So alanine side group ആയ amino ഗ്രൂപ്പിനെ അലാനിൽ നിന്ന് ആൽഫ കീറ്റോ ഗ്ലൂട്ടറേറ്റിലേക്ക് ട്രാൻസ്ഫർ ചെയ്യും. ഈ ഒരു പൊസിഷനിലേക്ക് ട്രാൻസ്ഫർ ചെയ്യും. So this become what? Glutamate, and this become pyruvate. So this is example for transferase, second enzyme. And third one is very simple. Hydrolysis. It catalyzes the hydrolysis of a substrate. So one of the important properties is what? It adds water. It's what? Add water here, water to the substrate, and simultaneously breaks them. There are three subclasses other. One is called proteases. So they break the peptide bond on proteins and peptide. So there are two classes under proteases. One is called peptidases and proteinases. So peptidases act on peptide bond adjacent to a free amino acid or carboxyl group. As you know, so this is an amino acid. There is one part is called NH2. You know that amino group. The other is called COOH. Clear? So this is a protein. So protein has two ends. One is called N terminal and this is a C terminals. So this act on what? Peptide bond adjacent to free amino acid here. Cut here from here like this, or carboxyl end from this end. ഇവിടുന്ന് കട്ട് ചെയ്ത് വരും, ഇങ്ങനെ കട്ട് ചെയ്ത് വരും. Okay, so they are called exopeptides. രണ്ട് സൈഡിൽ നിന്ന് രണ്ട് അറ്റത്ത് നിന്ന് കട്ട് ചെയ്ത് പെപ്റ്റൈഡ് ബോണ്ടിനെ ബ്രേക്ക് ചെയ്യുന്നുണ്ട്. ഇസ് കോൾഡ് exopeptides. Some of them act on the interior. അപ്പോൾ ഇതിങ്ങനെ കട്ട് ചെയ്ത് വന്നിട്ട് ഉള്ളിലെത്തുന്ന സമയത്ത് this becomes ഡൈപെപ്റ്റൈഡ് ഒക്കെ ആയിട്ട് മാറുന്ന സമയത്ത്, ഡൈപെപ്റ്റൈഡ് എന്ന് പറഞ്ഞാൽ രണ്ട് അമിനോ ആസിഡ് ചേർന്നത്, like this one amino acid, this is another amino acid, there's a bond here, this is one. Okay, so this is called a dipeptide. This is peptide bond, this is two amino acid. So this endopeptides act on this bond, this peptide bond, and breaks into liberate two amino acid. So this is a act on the interior peptide bonds of a protein like this. So such enzymes are called endopeptidases. And third one is the esterases, which catalyze the hydrolysis of ester linkage. As you know, the ester is the fatty acid തമ്മിൽ ലിങ്ക് ചെയ്യുന്ന ബോണ്ടാണ് ester linkage. അതിനെ തമ്മിൽ ലിബറേറ്റ് ചെയ്യും. Such examples are lipases, esterases, and nucleases are all examples for esterases. Then third one is called carbohydrates, which catalyze the glycosidic linkage of carbohydrates. Carbohydrates break it. For example, sucrase, maltase, etc. And next category is what? Ligases. The enzyme which catalyzes either the removal of a group of atoms from their substrate, leaving double bonds, or add groups to the double bond without hydrolysis or oxidation. Remember, there is no hydrolysis here, or oxidation, or reduction. So it removes what? Atoms, group of atoms like carbon, oxygens are removed, or hydrogen atoms are removed. So it leaves what? A double bond, double bond. So three important characters point is noted, should be noted here. One is called removal of group of atoms, and second one, as a result there will be a double bond. There is no hydrolysis, oxidation or reduction. Examples are aldolases, enolases, fumarases, etc. So they cleaves a carbon-carbon bond, or carbon-oxygen bond, or carbon-nitrogen bond. Okay. Isomerase, it catalyzes the interconversion of a compound to its isomers. This enzyme catalyzes the intermolecular rearrangement. One of the examples are phosphohexoisomerase. So this is called glucose 6-phosphate, and is interconverted to fructose 6-phosphate. Both are the same formula, but their structure are different. So phosphohexoisomerase convert the glucose 6-phosphate into fructose 6-phosphate. Glucose is a 6-carbon compound, and fructose is also a 6-carbon compound. So their structure are little bit rearranged. So this is called a isomerase. And the last one is called ligases or synthetases. They link. Ligases means what? Two compounds are linked. Catalyze the synthesis reactions by joining two molecules coupled with the breakdown of a pyrophosphate bond of ATP to ADP. So it's requires what? ATP is an energy consuming enzymatic reactions. So ATP molecules are used, and it is reduced to ADP. Examples are DNA ligases, RNA synthesis, etc. So one of the other examples are, so bicarbonate, both two are linked. A bicarbonate and pyruvate is combined together with the help of an ATP by consuming one molecule of ATP to form oxaloacetate or oxaloacetic acid. Oxaloacetic acid, which will be studied in the next unit. So bicarbonate and pyruvate are two substrates. Both are linked to produce what? Oxaloacetate by consuming one molecule of ATP. Okay, this is called ligases. And each enzyme has a classification number. Just remember it. This, for example, the enzyme hexokinase is number is 2711, 2 digits. 2 is the class here. Transferase, it comes under the group transferase. 7 is the subclass transphosphate. So what did it actually transfer? This actually means it's a transferase, whether it is transferring amino group or phosphate group. That is the number. 7 means what? Transfer of phosphate. And 1 is a subclass of subclass. ഇതിന്റെ subclass ആണ് അത്. Alcohol is a phosphate acceptor. Little more details. And the last one is what? Its specific name. ATP dehexo 6-phosphate phosphotransferase. Actually, the common name of hexokinase is, the common name of this enzyme is hexokinase. The real enzymatic name is what? ATP dehexo 6-phosphotransphorases.